All right, you guys, we're going to start activity 4, which are Ascomycetes. So you'll watch this lecture and there's some additional videos, and this will be for questions 8 through 11. And you guys are going to use the specimens for question 9 that are going to be provided at the end of this PowerPoint section. All right, so Ascomycota are sac or cup fungi. These are the largest and most diverse phylum. Their hyphae are septate, so they have cell walls. Many Ascomycetes and molds that there are many that produce asexually, and there are ones that do not have reproductive fruiting bodies. Molds usually reproduce asexually through spores called conidia. We'll talk about in a little bit. Yeast is a very important single-celled Ascomycete that lacks hyphae. It's really important in the world, but also especially for humans for its uses. So in general, Ascomycetes undergo sexual reproduction to form a fruiting body. The fruiting body is called an ascus. Asci is plural, and the ascus produces ascospores. Okay, so here are a couple examples of different fruiting bodies. So this is a cup fungus, and then morels are also Ascomycota, and then yeast, like I talked about before. All right, so some examples, you have different types of cup fungi that are really cool. They're usually pretty squishy. Black truffles are an Ascomycota, and then morel mushrooms, both true and false morels, are Ascomycota. Okay, kind of an unusual Ascomycete are these single-celled yeast. Yeast is found in many lineages, but the ones that we use that are most well-known, I mean, for making bread and beer and wine, are Ascomycetes. Their yeast is all around. You can find it on your skin, but you can also find it really frequently on the skin of fruits like blueberries and grapes. You can use it to make bread and wine, different strains of yeast. And its genome has been fully sequenced, and it contains 6,000 genes. It's a really well-studied model organism. Okay, so the life cycle of Ascomycetes. We're going to start here. At the beginning, once again, we have the 1N hyphae that are going to, just like we saw before, reach to each other via plasmogony, and then it will form these N+N dikaryotic mycelium that will grow and eventually become the mature spore-producing fruiting body. So the adult life is N+N. And then within that fruiting body, in the hymenium, still the sexually reproductive layer, karyogamy will take place. So the N+N dikaryotic nuclei will fuse to become a 2N zygote. And so that 2N zygote is going to undergo meiosis, and then it's going to undergo mitosis to form 1N spores. So before in Basidiomycetes, we only saw them undergo meiosis, so it only went from 1 2N cell to 4 1N cells, but because in Ascomycetes we have meiosis and then a round of mitosis, you always see 8 spores formed in the ascus, and those are 1N spores that are going to be spread around. Okay, I talked through this already, but here are the cells, here are the slides for it. Variations in form. In cup fungi, the hymenium is along the inner surface of the cup. And then in true and false morels, the hymenium is in the little ridges that look kind of like a brain. It covers the inner surface of the cup. So think of each little section as a mini cup. So there's hymenium layer within each of those. There's some other really cool examples of Ascomycetes. For example, this blue stain fungus is really common in the Blue Ridge Mountains. If you ever see a blue piece of wood, it's because this fungus is in it, and sometimes you can see the fruiting bodies of it. Okay, so the ascospores are developing within the hymenium. So if we cut into that cup fungi, we would see many asci, and you could see them at different stages. So remember I said initially the asci will be dikaryotic and N+N, and then will fuse together via karyogamy and form that 2N zygote. The 2N zygote will break via meiosis into the 4 1N meiotic cells, and then it'll go through mitosis to go from 4 1N cells to 8 1N cells. And so a mature asci has 8 ascospores in it. So you can tell when it's hit its peak maturity when it's gone through both meiosis and mitosis, which means it's gone from 2 to 4 to 8 cells. So when there's 8 ascospores in an asci, it is mature. Okay, so you can see that they're stained purple on this slide. So you can sometimes, once they open up at the top, some of them might fall out, but you can definitely tell there's more than 4. Here's what you're looking for. So 1, 2, 3, 4. There's more than 4 within this individual ascus. So that's really characteristic of Ascomycota, is that they have 8 ascospores per ascus. Okay, see that pretty clearly here. All right, so within a morel, if you were to cut into it, you would see the hymenium layer along those inner ridges, and the hymenium layer, just like we saw before with the basidiomycetes, is really high surface area all within here, and then there are asci that will divide into ascospores on that hymenium layer. Okay, so asexual reproduction is more common in Ascomycetes. Asexual spores are called conidia, and they are formed at the end of specialized hyphae, which are called conidiophores. Each mating type, for example, a plus and a minus, could reproduce asexually by undergoing mitosis to form haploid spores. So remember, it's an exact copy of the parent organism. The conidia and Ascomycetes are in long, skinny chains on the conidiophore that branch off of the hyphae. So remember that when we were looking at zygomycetes, they looked more like a solid lollipop, and these ones look like they have long chains to them. Okay, so the conidia are the things that would spread. They'd be one end, and what would be a copy of the parent cell that would be spreading around. They're contained within this vessel, which is on a stalk called the conidiophore. Okay, it shows you that again. Sometimes the chains can be pretty small, sometimes they're larger. Penicillin, which is forms for penicillin, is a ascomycete as well. It forms some it reproduces frequently by asexual means. Okay, so here's some videos about ascospore spore dispersal. Pretty cool. And here are the specimens that you will need to answer question 9.